Well, hello everyone. It has been a whirlwind of a week. I'm sure it's been the same for you too. We are going to start mutations and chromosomes and karyotypes, which is kind of the last part of the DNA chapter. Once we take a look at the structures and the transcription and translation process and DNA replication, we want to take a look at some of the things that DNA does on a larger scale, or how it looks on a larger scale. What happens if it goes wrong, if there's hiccups in those A's, T's, C's, and G's, what are the potential causes, as well as if you have more than the number of chromosomes than you need, or if you have less than you need, what kind of disorders and difficulties can arise from that. Now, before I do that, I have to show you the newest addition to our family. This is Rhett Jacob Tierney. Everett Jacob Tierney is his name, but we are calling him Rhett, and this is the reason why I've been gone for the last couple days. So just wanted to show you the new addition to our family. Mama and baby are doing great, and so he's bright-eyed and bushy-tailed, and looks a little frustrated at you guys. He's wondering why haven't you guys kept up with your reading like you're supposed to. Just kidding. Okay. Now the other thing I want to bring up is a lot of you had questions about your summaries that you needed to submit on Turnitin, and I had a lot of emails that didn't work very well, and some of you sent me attachments in email. Please don't fret about that. We'll get all that straightened up when I get back. I am going to take a paternity leave this week, and so you will have to. We're going to look at microscopes this week and do a little scavenger hunt. So I will not be there for this week. So please help each other, and I just hope that this is a successful week for you. All right, let's finish the last part of DNA. Is that okay with you, Rhett? He says it's okay. So, the first thing we need to do, this objective up here, sorry for my notes, they look a little bit weird. That's because the formatting, there's errors in it when I copied this PowerPoint over, so bear with me. We're going to explain mutations in terms of something called a frame shift mutation and a point mutation, and I think once you kind of look at the word frame shift and you see an example, it's going to make sense to you. But first, what is a mutation? Okay? A mutation is just a change in your DNA. Now, when you hear about mutations, you might think of words like mutants, or just, like Teenage Mutant Ninja Turtles, or you might think of something as a mutant and it probably has this adverse effect. It looks odd, or it doesn't look the way that it's supposed to. It might be neon green, or glow, or be a life-size karate fighting turtle. But I want you to know that DNA mutations also occur randomly, and every one of you is also a mutant, because you have A's, T's, C's, and G's that are different now than they were when you were born. And this can be just because of mess-ups when that DNA replication happens and DNA polymerase binds. It's really good at matching A's with T's and C's with G's, but every once in a while it will make a mistake and it won't correct that mistake, and so you have a different base pair than you originally had when you were born. Okay, so mutations usually do occur during DNA replication, and it would occur during this phase again, because remember, that's when we're building new DNA, and that's when A's are binding with T's and C's are binding with G's, making more from the original template. Now, can you also get mutations because of other reasons? Absolutely. Anytime that you're sun tanning and you have those nice UV rays that are, warming up your body and turning your skin all nice and tan, well, that looks really nice, but UV radiation, that wavelength of light is just about the same size as your DNA. So when you have UV light that comes into the cells of your skin, it can go straight through your cells and straight through your nucleus and actually hit your DNA, and sometimes it will cause those A's, T's, C's, and G's to change. So the more time you spend in the sun, the more fair complected you are. If you got blonde or red hair, if you got freckles, and you don't put that sunscreen on, then you are more susceptible to the effects of the sun, skin cancer, UV radiation, that kind of thing. Luckily for other people, not myself, I burn pretty easily, but if you have dark hair, if you have brown eyes and you're kind of dark complected and you just tan, well, lucky you, because usually if you have darker complexion skin, that means that your skin protects you more from UV radiation. Now, as a little side note, what I will say is there's a trend. You can actually see how skin color changes with the change in UV radiation intensity on the globe. So what I mean by that is the further north and south you are from the equator, if you live there, people in those areas tend to have lighter color skin the further away from the equator that they live. I don't know if you've ever thought about that, but you think of people that live in like Norway or maybe in the southern parts of South America, or they live in Australia and New Zealand. The further away you get from the equator, usually the lighter complexion you are, because you don't need that protection. However, a lot of people that live right around the equator have browner, darker skin, and that helps with UV radiation where the sun is much more intense. All right, moving on. So, first category of mutations are the less severe kind. These are called point mutations, and point mutations are kind of just like they sound. They are changes in one base of DNA, and it's only in one position. Sometimes we also call them substitution mutations. This can have the possibility to change an amino acid or not, depending on where it's changed. So, for example, you can have an amino acid that has a codon of UGG, let's say, and if there's another codon for that same amino acid, if you change one of those bases, you might still end up with the same amino acid even though you changed one of the bases in the codon, and you won't notice anything. It'll just act like the same exact protein. So let me show you here. Let's say if I wrote a sentence here, this is going to be kind of messy because unfortunately I don't have a stylus with me, but I'm going to give it a whirl. So let's say I have the cat, this is bad, isn't it, ate the bad H, oh man, look at that, sorry, the dog, okay? I'm not going to get that dog in there, am I? Alright, the cat ate the dog. Excuse my writing. A point mutation, if these are all three-letter words, and I made them three-letter words because of they're kind of like codons, right? So let's say this is a normal sentence, and if you were to make a mutation, well, what would happen is just how I drew these words in here. Let's say I messed up this letter right here, and instead of an E, there was a mutation that occurred at that specific point, and instead of an E, that was substituted. Again, these are called substitution mutations also, where this E used to be. Instead, let's say that we put an A, okay? Now, if you saw this, you'd say, well, geez, that's kind of a messed up sentence, but I still think I get the meaning of it. I think that this right here is supposed to be an E, but it looks like an A, so I think they misspelled the word the, but I can still get the cat ate the dog. It still makes sense. And that's a lot of times what your DNA does. You might have one base that changes, but your DNA still isn't really harmed that much. It still does the same thing it's supposed to do because all the rest of the letters are in the right spot. Hopefully that makes sense. Now, let's talk about a worse kind. So, this is another example. I probably should have just showed you this instead of drawing my awful drawing. But if you take a look right here, The first codon is AUG, which we all know is this methionine amino acid for the start codon. And then GCC codes for alanine, if you look on that chart that I gave you. Now, what you notice here, this is a substitution mutation, and then you see this word off to the left here that says silent. It's a silent mutation. Well, why is it silent? And if you take a look, GCC and GCT, even though this substitution happened right there, the C got switched for T, luckily GCC and GCT both code for alanine. So you'll notice this protein chain, this polypeptide, and this new polypeptide with the mutation, they're the exact same. There would be no effect when this mutation was made. And you keep making the same protein, it would be perfectly fine. And again, you wouldn't know it, and this is again why we call it a silent mutation. Now, other times with hemophilia, for example, on the video, that girl who had not hemophilia, excuse me, sickle cell anemia. Sickle cell anemia is caused by one of these frame shifts, and so what that does is it causes a not frame shift, excuse me. It's caused by a substitution mutation, and alanine is switched with a different amino acid, let's say. And what that does is it alters the shape of the protein, and it is not silent because she has a disorder. I mean, that misshapen protein causes her blood cells to look different, to not flow as easy, to not carry as much oxygen. So sickle cell anemia is just caused by one base pair difference, one substitution. Alright, second category here. So let's say, I'm going to go back to drawing even though there's something on the next page. Sorry for the bad drawing. Let's say you have a letter that doesn't get substituted, but it just kind of shoves its way in there. These are called frame shifts or frame shift mutations, where one base is actually inserted or it could be removed from the DNA. It's not substituted, it's not traded in and out. It's just either added or it's subtracted. So that's the big difference. And what this does is it changes the reading frame of your RNA polymerase, and you make different codons happen. And different codons mean different amino acids, and usually this results in a very big interrupted mistake. Okay? Now, these fall into the same category right here. So insertions, these are called, this means just you're inserting a base. Deletions mean you delete a base. Actually, in the science world, we just kind of say that we combine these two together, and a lot of times you'll hear the word indel mutations. Indel mutations. In for insert, del for deletion. So these are called indel mutations. All right, let's see how well my fingers work this time. If I'm going to put here the cat ate the dog. Oh, that was a bad G. Dang, I was doing good. Okay, the cat ate the dog. Same sentence. If I was to add a letter, like let's say I'm going to add a letter right here between the C and the A. It's an insertion. Okay, I'm going to add it. Well, what that does is it moves everything down one frame, or a frame shift, if you will. So how this results is if I was to add a new base right here, let's say I add an H, okay? Well, if I add an H, this is what's going to happen. It's going to move all the other letters down, and we're going to get something that looks like this. Are you ready? Watch my bad writing. So there's a C, but we're going to add this H in here, okay? And then we have to pick up where we left off. So where this H got inserted right here, well, what would come next? This A would come next. And so what you would see next is something like that. And then your next three letters, if you follow it along, are T-A-T, and then E-T-H, and E-D-O, and then a G. So I'm going to try and write that again. So I'm going to see here. H-A, so that's going to be T-A-T, and then E-T-H, right? T-A-T, E-T-H-E-D-O. And then a G. Now this looks a little funky, so sorry, but if you look at that bottom line, just because we added this H to the mix, this no longer is the cat ate, this no longer is the cat ate the dog. It's the cha tat eth edog. And hopefully you're like, what the heck does that even mean? Well, it means that it's such a bad mistake that usually there's a disorder there, or it might mean that a fetus doesn't even develop. It's a lethal mutation, and it moves the frame of those 3 letters down by 1. So again, that's why they call this a frame shift mutation. Hopefully this will make more sense on the next slide. Here we go. I'm sorry for this being cut off. I'll try my best here. Let's see. So, even though this is cut off, I'm going to try. Notice right here that this says ATG and then GAA. Well, let's say that G was taken out. Now, what I drew on the last slide, that I added. That was an insertion. What they're showing you here, this is a deletion. So this is a deletion frame shift mutation. Well, if you take a look, ATG is fine, but what if we got rid of this G? What would the next 3 letters be? Can you guess? If you got rid of the G? Hopefully you had time to think, and it would be AAG. And that's what you start to see down here. Well, AAG does not code for this amino acid. AAG codes for something else. Your next 3 letters would be CAC, and that's what you see down here that's cut off, and then GT. So it moved everything over this way. It moved everything over actually kind of to the left. If I can put an arrow in there, just by taking this G out there, it switched everything back 1 frame, or a frame shift, and it caused all the amino acids in the chains to be deleted. So those are mutations. If you have any questions, please email me. I'm more than happy to email students when I get chances this week. All right, let's talk about chromosomes. When I was in high school and I saw a chromosome, and usually chromosomes just look like X's, kind of like this, I'm like, how the heck is that DNA? I don't get it at all. And my biology teacher never told me. It was just like, the DNA is double helix shaped and these are chromosomes. And I'm like, well, I don't understand how that works. So hopefully this will make some sense. First off, let's take care of the nitty-gritty. Chromosomes are DNA that is bundled into tiny, dense packages. This only exists during cell division. So when the cell's not dividing, it doesn't even look like this. It's kind of this stringy mess called chromatin instead of chromosomes, which we'll talk about. So these only exist right before the cell is getting ready to divide during mitosis or meiosis, which we'll talk about a little bit later. But usually it doesn't look like this. Usually it looks like spaghetti, honestly. So anyway, the center of the chromosome, this is something I will definitely ask you on a test. This guy right here, this is called the centromere. The center of the chromosome is called the centromere. Here's just another picture to kind of show you that DNA is wound up really, really tight in these kind of bundles here, and so it forms basically this X shape. Okay, so let's start before chromosomes. Let's talk about chromatin. Now, it's hard to keep all these words straight because they all kind of sound the same. Chromosome, chromatin. Chromatin is the unpackaged DNA found in the nucleus when the cell is not dividing. It kind of looks like spaghetti. It's all unraveled. It doesn't need to be all compact into chromosomes because it's not going to try and split all that into 2 cells during cell division. This is where the DNA is wrapped around proteins. These proteins are called histones. You'll see that on the next diagram. And it looks kind of like beads on a string. I called it spaghetti noodles, but yeah, they kind of look like beads on a string. So let me show you. So this is an electron micrograph, which is basically a very tiny photograph using electron capability. And what you see here is just this little stringy stuff, okay? But then you see these little white dots right here, and these are proteins where this DNA is just wrapped around really, really tight, and it's just continuing to do that. The DNA just continues to wrap and 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 wrap. So that's chromatin. This is how your cell's DNA exists. When? When do you have chromatin, or when is it all laid out? Hopefully you said when the cell is not divided. So this is more how your DNA turns into a chromosome. If you look up at the top here, this is your double-stranded double helix. All right? Now right here, this is just your regular DNA strand, short region of a DNA double helix. Well, then if you take a look at these brackets, it shrinks this down, and this image that you see up top here really just exists right here. Now those beads on a string I was talking about, these are those yellow guys, and again they're called histones. But you'll notice now this red DNA just wraps around each of these histone proteins. Then, after that, it gets wrapped again, and it kind of wraps over on itself. And then if you look at that, it wraps over on itself again, and then those coils wrap, and then those coils wrap, and they're actually called supercoils. So it's kind of tough to imagine, but this is what I try and tell students. If you've ever been on a swing and your friend was maybe pushing you, your mom or dad was pushing you, or maybe you were doing it by yourself, but then on that swing you started to twist in the swing, and the chain started to swirl, right? And then you twisted some more, and then you twisted some more, and you kept on going. If you've ever noticed how the chain doesn't just turn into a spiral after a while, the tighter you wind it, it kind of doubles up on itself, if you've ever experienced that. Like the chain just isn't spiral shaped, it gets so tight that it even doubles up and starts to coil even more on top of that. That's what we're talking about, that it's so tightly wound, and it's wound over and over and again on itself that it just continues to kind of look more and more compact. But at the heart of this, you see this kind of bigger image. Well, this will eventually turn into an X shape, which is your chromosome. But it's all this just tightly wound DNA at the smallest level. Moving on. Here's another picture just to kind of show you. DNA strand that gets wound around these histones that are cut off, but these get wound right here. And then those histones, that coils more, and then that coils more, and eventually it coils into these kind of left and right sides. Now what I haven't showed you here is that here's that centromere that you took notes on, but this side and this side are identical. You have one copy of your DNA. This right here is a second copy of your DNA. Now maybe you could guess this, but if you know that this is one copy, and you know this is an exact different copy, they're two of the same copy of DNA, and cell division happens, what do you think is going to happen to this guy? Well, I'll let you think. Hopefully you said maybe something will cut it right there, right down the centromere. And what will happen is that this copy will go this way, and this copy, oops, sorry, not very good at this. This copy will go that way, and this copy will go this way. And they'll each go into their own cells. This one will be a cell over here, and this one will go into a cell over here, so that both of them will have a copy of the DNA that goes into that cell. So this happens during DNA replication, just to let you know. When DNA replication happens, it starts off as this, and then it creates a sister part of it, or an exact copy of the same chromosome. This is something that I just wanted to say real quick. Let me go ahead here. There's one copy right there, and this is an exact copy of DNA on this particular chromosome. Now, something you will definitely be asked on a quiz. Humans have a total of 46 chromosomes. These are all paired up with another chromosome, so we say you either have 46 chromosomes, or more likely you have 23 pairs of chromosomes. Now, why do you have pairs? Maybe you want to think about it for a second. Why would you have why would you have 23 pairs of chromosomes, or, have two chromosomes that are the same? Well, hopefully you might have said you get one from your mother, you get another from your father. Now, to be a little bit clear, it's not just one. You get 23 chromosomes from your mom, and you get another set of 23 chromosomes from your dad, to add up to a total of 46. All right, analyze a karyotype. So what this is, I should have said that on the last. Okay, sorry for the hiccup. On this page, what you see here, this picture right here, we call this, and it's kind of in dark, but it's called a karyotype. So practice saying it so you know how to say it, karyotype. And what you see here, these are actual human chromosomes that somebody was able to isolate from a cell nucleus, and he laid them all out on a microscope, and he actually took microscopic pictures of them, which is pretty darn sweet if you ask me. Now, if you notice, this person has two first chromosomes, 2 second chromosomes, 2 third chromosomes, all the way down the line, and they're all numbered. Notice you have 22 pairs of regular chromosomes, but then your 23rd pair is not labeled the same. And if you look right here, this 23rd pair is very special. It's what makes you a male or female. Now, is this a male or is this a female? Let me just ask. If you have an X you have an X as one sex chromosome, and you have a Y as your second sex chromosome, X and Y, what are you? You would be a boy. All right? Y chromosomes are a lot shorter, and actually, boys, we get a bad rap in the science world because that's why females say that boys are stupid, which we're not, just to let you know. Okay, but they say, well, since you guys only got this little bitty bit of DNA on the Y chromosome, you don't have as much information in our bodies. Well, whatever. Okay, so a karyotype shows the complete pairs of chromosomes arranged in order of decreasing size. So I didn't say that yet, but if you take a look at this, your longest chromosomes are your first chromosomes. And then if you notice, these are a little shorter. Your third pair is a little shorter than that. Then your fourth pair, and your fifth pair, and your sixth. Then if you notice, you'll see a trend as you go down the chromosome numbers, how short they get. So this is a human karyotype of a boy, or a male individual. Now let's take a look at some problems. What happens if you have a mistake with a whole chromosome? Not just a mistake in your A's and T's and C's and G's, but you get a lot of extra DNA, or you're missing a lot of DNA. When that happens, it's kind of like information overload, or there's too much information that's lost. And a lot of times, if you're missing a chromosome, or you have a chromosome that's been added, in other words, you have 3 chromosomes at any given number instead of just 2, most of the time that will be fatal, unless it's a relatively small chromosome. So what you see on this first example is something that we call, I've heard it pronounced trisomy or trisomy. I kind of tend to say trisomy because it sounds like chromosome. But trisomy is a disorder where you have 3 chromosomes instead of your normal 2 at each spot. Now, trisomy 21, that means that instead of 2 21st chromosomes, some person would have a third 21st chromosome. And you might not have heard of trisomy or trisomy 21, but I'll bet you have heard of a syndrome or a disorder that we call Down syndrome. And Down syndrome is because of the person having one extra chromosome one extra 21st chromosome in every single one of their cells. So, most of these, most individuals with Down syndrome will survive, but depending on how much of that chromosome they got, it's kind of an inverse relationship in the sense that if they get just a tiny bit of that third chromosome, well, that's not too much extra information, so they will still have the syndrome or the disorder, but they will be very high functioning. They might have jobs, they might live on their own, they can carry out very nice conversations, but if another person with Down syndrome, if they get the entire third chromosome, that's a lot of information that your body can't handle. And so those people with Down syndrome that acquired the entire third chromosome, they tend to be lower functioning, where they need a lot of, they need constant care, and usually cannot live on their own. That's usually 1 in 900. By the way, this number goes up rapidly the longer females wait to have kids. Like if you're over 40, the number decreases to like 1 in, I think, 200 something. You'd have to look that up. But it decreases pretty rapidly the older you get, especially after age like 35 or 40. Okay, the opposite of that is called monosomy. Mono meaning one, that's the prefix. And this is the exact same thing, only instead of adding a chromosome at a given site, we're getting rid of a whole chromosome at a given site. So the example here is something called Turner syndrome, where this is actually a sex-linked syndrome. In other words, you only have one X chromosome. And so somebody who has Turner syndrome would be considered female, but they would also be considered sterile. And there's other complications that would go along with that. Let me show you a karyotype. First off, hopefully you can see which karyotype this is referring to. This person has 23 sites. If you count them all up, there's still 1, 2, 3, 4, 5, 6, all the way down to 22 plus the sex chromosomes. But if you notice right here, there is a third 21st chromosome. This is trisomy 21, or this is a person with Down syndrome. The next question I'm going to ask you, is this a boy or a girl with Down syndrome? So take a look, make your guess. Hopefully you said this is a female. And the reason why that is at the Y spot, you see that there is no Y chromosome. However, this individual does have 2 X chromosomes. Let's look at Turner. This is kind of an odd looking karyotype. Again, everything else looks in order. Your first, second, all the way down to your 22nd chromosome. All those chromosomes look just fine. But if you notice, there is 1 X chromosome and no Y. And so you're missing quite a bit of information here. And those individuals that have only 1 X chromosome, again, they are considered female, but they will have a lot of other side effects. And one of those being from the last slide, they will be sterile individuals. They will not be able to reproduce. And rightly so. If they only have 1 X chromosome, then they don't have a chromosome to donate to their offspring. So, that's all I have for you. I hope that you enjoy this. I hope you have a good week with microscopes. Be patient with the microscopes, because sometimes, let me move ahead because it's a chart showing you how many survival, I should say, how many individuals survived with different trisomies of different chromosomes. Now, you see trisomy, these all represent chromosome number, and if you remember from a couple slides ago, your first chromosome is your largest chromosome. Well, if you take a look at this, there were 85,000 live births. How many of those were living? Well, you have 0. And you have 0 for 2 and 0 for 3, all the way from 6 through 12. Again, 6 through 12 right here. So, what does that mean? It means either they were spontaneously aborted or there were no live births. So if you take a look at this, 561 plus 95 plus 53 plus 159, none of those individuals survived. The trisomy, the third chromosome, was too large, it was too much information, and a fetus didn't develop. That's what we're saying. Now if you notice all the way down here, this G bracket, here's that 21st trisomy, or trisomy 21, so we notice this is Down syndrome. And we have more live births, just to let you know. There are still some abortions here, some spontaneous abortions. By the way, spontaneous abortions, this means like a miscarriage. So, but if you notice, there are more survival. There's a higher survival rate the lower that you go in your chromosome number. I should say the higher you go in your chromosome number, but those chromosomes are smaller. You'll also see some other syndromes here, like you could have 2 Ys, or you could have 2 Xs, or just like Turner syndrome, you could have an X without another chromosome, which is represented by that circle. You could have 3 X chromosomes, or this is called trisomy X, or trisomy X. Some species of plants can be polyploid, where they could be tetraploid at every site. They could have 4 chromosomes at every single location, which is actually okay in plants. But in humans, we need 2, 1 from mom, 1 from dad. Anytime you have an extra chromosome or you have 1 less chromosome, usually that's pretty devastating genetically. Oh, that's it. I hope you have a good week. The microscopes can be a little bit challenging. Please read through your microscope tutorial from cover to cover, because it will definitely help you out with using the microscopes and the slides. Make sure my lab's perfect when you're done, like it always is supposed to be. And miss you guys, but I'll be back in a week, and I hope that you guys have a good one.